Welcome friends, we will discuss in this part monohybrid cross. So far we have discussed Mendel's experimental material, that was pea plant. We have discussed Mendel's experimental technique. We have discussed selection of parents by Mendel. Today we will discuss the crosses performed by Mendel. Today's topic is monohybrid cross. Mendel's monohybrid cross. Monohybrid cross. Monohybrid cross. It is also called as inheritance of one gene. Inheritance of one gene. What does this inheritance of one gene mean? I will just explain you a little while after. Inheritance of one gene. Inheritance of one gene. What does this monohybrid cross actually means? A breeding experiment, if I will tell you, a breeding experiment, right? Dealing with a single character. Dealing with a single character is called as monohybrid cross. Monohybrid cross. Monohybrid cross. A breeding experiment or a cross. Other name of the cross is the breeding experiment, right? A breeding experiment or a cross dealing with a single character at one time, I may say at one time, is called as monohybrid cross. Mendel performed many crosses. He performed many crosses. He performed monohybrid cross. He performed dihybrid cross. He then performed trihybrid cross. But he performed these crosses in a very systematic way. Those biologists who tried to perform breeding experiments before Mendel did not get a good success. The reason was that they performed crosses, but those crosses were taking into consideration many characters at a time. Whereas Mendel performed breeding experiments in which he took into consideration, in which he involved characters one by one. In one experiment, he just took consideration into height of situm. In other experiment, then he took another character. Then he took two characters, height of situm and color of flowers. So he was in a position to get the clear-cut principles, laws, which laid the foundations for the modern genetics. Okay. This was the hallmark of Mendelian work. He did experiments in a very systematic way. Whatever he did, he kept a good record of his observations, right? Whereas his co-biologists who tried to find out the inheritance principles or laws before him, they failed because they wanted the complete inheritance of an organism at a single time. But Mendel did reverse of that. Sorry. What he did, he took consideration one character at one time. Then next cross, he took into consideration two characters. That is dihybrid cross. So this is monohybrid cross, right? Monohybrid cross, a breeding experiment or a cross dealing with a single character at one time. This is known as monohybrid cross. If two characters are dealt with at one time, that is dihybrid cross, right? So what Mendel did, he crossed pure plant, he crossed pure tall plant with pure dwarf plant. And he took into consideration one character consideration. The character was height. The character was height. With its two alternative forms, tall and dwarf. Tall and dwarf. But यहाँ पे आपको एक बात जरूर याद रखनी है. That's Mendel crossed a pure tall plant. I would have said you that he took a tall plant. There is a huge difference between a tall plant and pure tall plant. When I say he took pure tall plant and pure dwarf plant, it means that these tall plants, which were used as parents, which were selected as parents by Mendel, these were true breeding. Because when they were inbred, when they were self-pollinated, they produced only tall plants. If tall plant was crossed with a tall plant, it produced only tall plants. Right? If dwarf plant was crossed with dwarf plant, it produced only dwarf plant. Then and only then, when he was sure that a tall plant on self-pollination, self-crossing, self-healing, self-fertilization produced tall plants, then and only then he selected this as a parent plant. And in fact, it took him 2 years to ascertain whether a plant which he was supposed to select as a parent breeds true. It took him 2 years. I will be exact in giving you the information. It took him 2 years to make it sure, to ascertain that the parent selected by Mendel was pure. When I say pure, was true breeding. When I say true breeding, that means that the offspring is produced by this plant were similar in phenotype and genotype with their parents. Okay? So, he crossed it. Let me write down here. He crossed it. Mendel crossed it in monohybrid cross. What he did? He crossed a tall plant, right, with a dwarf plant, right? Dwarf plant. And he selected them as parent 1 and this was parent 2, right? This was tall plant, but pure tall, true breeding, right? This was dwarf plant, but pure dwarf. True breeding dwarf, right? And these parents, right, tall plant and dwarf plant, they are presently called as parental, they make parental generation presently, and they are represented by symbol P. Okay? Presently they are designated by symbol P, parental generation. Though Mendel was not of this, vocabulary that he used parents as parental generation. These were later on modifications made by other biologists who were interested in carrying forward the work of, Mendel, right? So, when he crossed two plants, pure, one was tall, pure tall, and other was pure dwarf, right? What he did, he got seeds. What is this crossing? This means that this fertilization between them takes place, self-pollination. Pollen grains are transferred from this plant. Here they mix with, lying down, stigma of corbel. Then pollination takes place. Then ultimately, fertilization takes place. Then seeds are formed. Those seeds were then sown by Mendel. And the plants raised from those seeds, those were called as F1 plants. What were those called as? F1 plants. Right? F1 plants. When this was crossed, how did he do this thing? Here you see, these are the gametes. One gamete of tall plant, right? Here also gametes. One gamete and one gamete. There are two gametes and there are two gametes. Right? Then he crossed them. Then he, I have already told you in my previous, video lecture, then Punnett square. How we can represent this by a Punnett square? Right? This is like this. Then came the, from this plant, tall plant, came the gametes. Again, here came the gametes from this plant, dwarf plant. These were the gametes. Then these were, fused with each other. These gametes were fused with each other by a Punnett square. Like this, you can see. I have already told you about Punnett square. You might be knowing everything about Punnett square. These were male. Suppose this was male, right? These were female on the vertical row. There is no binding. While you can write either on horizontal, row or vertical column, like this. You can see here T. Then on fusing we can get result like this. T and T. Then this T. And this T. These plants, 1, 2, 3 and 4 plants, these were called as F1 plants. What he got, all the plants in F1 generation, these F1 plants, this was called as F1 generation or F1 generation or first filial generation by Bateson and Sanders in 1902. First filial generation or F1 generation, right? All the plants in F1 generation, right, were tall plants. Now, if I will tell you the ratio was tall plants. All the 4 were tall plants. There was none of them was dwarf. Then if I will write down here ratio, phenotypic ratio was 4:0. Right? Phenotypic ratio. Phenotype. All were tall. Tall. And genotypic ratio for them was same, 4:0. Tall. All were heterozygous talls. Right? Hybrid tall. Phenotypic ratio, when I look the expression, general appearance, this is phenotype. When I look for the genetic makeup, then these are the genes. These all plants in F1 generation were hybrid tall plants or heterozygous tall plants. This is a tall plant, this is dwarf plant. Now we have a tall plant. T and T, right? Small t. This is a tall plant and this is tall plant also. This one. What's the difference between this tall and this tall? Right? This tall is heterozygous tall or hybrid tall. This tall is hybrid tall or heterozygous tall. While as this tall is pure tall. What does this mean? This means that homozygous and heterozygous on that homologous chromosome. This tall plant, right? Heterozygous tall plant. The two alleles of a gene were unlike, corresponding loci in homologous chromosome. These two alleles were unlike. Hence, this is known as heterozygous or hybrid tall. On homologous chromosomes, on corresponding loci, corresponding positions, this plant was having two alleles which were identical. I have told you already in my previous video lecture what is a homozygous and what is heterozygous. I am just repeating here. So, in first generation, F1 generation. First, it was the parental generation, P generation, parental generation, simple P. Then when these parents were pure, when these were crossed, right? They produced plants. Those plants were all tall plants. But only difference between the parent tall and the F1 generation tall plants was that F1 generation tall plants were hybrid talls. They were heterozygous talls. While the parent was the, right? This was homozygous tall. Okay. Then came the second generation. Now we have like this. Let me rub it. This was the tall plant. It was crossed with dwarf plant. We got F1 hybrid that was like this. F1 hybrid. This is hybrid tall. This is also called as F1 hybrid. This is also called as F1 plant. And the generation of this plant, right, was known as F1 generation or first filial generation. Filial generation. Because filia means daughter and filius means son. Right? Latin words. Okay? Then, what he did, he just self-pollinated these plants. Self-pollinated. What he did? He self-pollinated these plants. Suppose this is a tall hybrid plant. Take a hybrid tall plant. It was crossed, self-pollinated with another same plant. Right? This is known as self-pollination. Tall plant, hybrid tall, when crossed with another hybrid tall. Right? This is known as self-pollination. When they were self-pollinated, seeds were sown, right? Plants were raised from those seeds. And the plants which were raised from those seeds, they formed the F2 generation. Right? What they formed? They formed F2 generation. How they formed? Let me make a Punnett square. Here we can see, this is T, one gamete, this is another gamete, right? Of this hybrid tall, and this is also T. One gamete, this is another gamete. Now, Punnett square, you see Punnett square, like this. Punnett square. You just see, this is the Punnett square, right? यहाँ पे आप देख सकते हैं, this was male and this was female. T and T. T and T. These two gametes. Right? These are male gametes. Right? And these, were written on the horizontal row. While these were female gametes, these were written on the vertical column. What the result was? This was like this. T, 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 and T, T. Here, this is F2 generation. Let me write down once again because there may be some camera problem. Right? Now this was the tall plant, this was dwarf plant. When they were crossed, they gave this hybrid tall. Now these hybrid tall plants were self-pollinated. Right? Accordingly, they give two types of gametes. Here this is one gamete, here this is one gamete for dwarf, this is one gamete for this, this is one gamete for one gamete and one gamete, one gamete and one gamete. These were now self-pollinated, like this. Okay, this is male gamete. Write down these as male. These are female gametes. Write down these as female. Now fuse them. TT, 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 and TT. These plants form F2 generation. Now, from F1 generation, by self-pollination, F2 generation was raised. Right? Now see the result of F2 generation plants. There were plants like this. TT, it's tall. Right? One tall. Okay? Then TT, these were 2, 1 and 2. These are tall. Right? But they are 2. 1 is to 2 is to 1. Right? This was 1. Dwarf. Okay? This was dwarf 1. Then their genotypic ratio was 1 is to 2 is to 1. Genotypic ratio. When I take genes into consideration, this was 1 because both the alleles of a trait were similar. Then it was pure tall. Pure tall. This was also tall. These were 2, 1 and 2. But these were hybrid talls because alleles of a trait character were dissimilar. And then this was, dwarf. This was 1. Then 1, 2, 1. This is genotypic ratio. What was the phenotypic ratio? Actually, the most importance is of phenotypic ratio. Phenotypic ratio. Okay? This was 3:1. 3:1. 3 plants were tall, right? Whereas 1 plant was dwarf, right? 3 plants were tall, 1 plant was dwarf. Then the phenotypic ratio was 3:1. What was genotypic ratio? Genotypic ratio was 1 is to 2 is to 1. How can you differentiate between a phenotypic ratio and a genotypic ratio? When I say phenotypic ratio, that means I say tall, and dwarf. Right? यहाँ पे 3 plants tall थे and 1 dwarf था. This is phenotypic ratio. When I say genotypic ratio, if there were 3 plants tall, out of the 3, how many were pure talls and how many were hybrid talls? One was pure tall because both its alleles were similar. I have already told you. And 2 were hybrid talls, heterozygous talls, because the 2 alleles of this, trait or character, they were dissimilar, unlike. So this was genotypic ratio. 1 is to 2 is to 1. And the phenotypic ratio was 3:1, 3 tall and 1 dwarf. And this is known as Mendelian monohybrid ratio. This 3:1 is also called as Mendelian monohybrid ratio. Okay? When in the first setup, 2 plants, one was tall and other was, dwarf. When they were crossed, all the plants of F1 generation were talls, hybrid talls, right? But when these hybrid talls were self-pollinated, then the ratio of tall is to 1, this dwarf was 3:1. So in the first generation, all plants were tall. None was, dwarf or intermediate in size in F1 generation. When these F1 generation plants or F1 hybrid plants, they were crossed, self-pollinated, right? They gave a combination of plants in which 3 plants were tall and 1 plant was dwarf. It means that in F1 plants, there were characters for both tallness and dwarfness. But in F1 generation, these plants failed to express the dwarf character. But in F2 generation, these characters were expressed. Right? Because out of 4 plants, 1 was dwarf and 3 were tall plants. So we obtain plants in a ratio 3 plants tall to 1 plant dwarf. It means that these tallness and dwarfness, which became evident which we saw in F2 generation plants, right? This was already present in F1 hybrid plants. But in F1 generation, these plants failed to express. This is known as law of dominance. Here the character 1 expressed itself, that was tallness in F1 generation. But the other character suppressed itself. It showed its effect in next generation. Right? And then, in the same way, these plants were then, F2 plants were used to raise F3 generation, F4 generation, so on. And in all generations, Mendel got the result 3:1 plants. 3:1. 3:1. So, what we have, learned today in monohybrid cross, we have learned that in F1 generation, the phenotypic ratio is 4:0. Genotypic ratio is also 4:0. Because all the plants in F1 generation were tall plants. None was dwarf. Okay? Then in F2 generation, right? F2 generation, the phenotypic ratio was phenotype. I am talking of phenotype. 3:1. 3 plants were tall, 1 was dwarf. While as genotypic ratio was 1:2:1. 1 plant was pure tall, 2 plants were hybrid talls, and 1 plant was dwarf. This is genotypic ratio. Sometimes it may be asked in the board exam, right? So, this phenotypic ratio was 3:1, which is also called as, Mendelian monohybrid ratio. In this way, Mendel, showed the results of monohybrid cross, which he conducted. Then he conducted the dihybrid cross. Okay, let me summarize some of all, events of, monohybrid cross. Just what was done in monohybrid cross. Tall plant was crossed in, with dwarf plant. In F1 generation, these plants, all plants were hybrid tall plants, right? When these hybrid plants were self-pollinated to raise F2 generation, the percentage was 3 plants were tall, right? And 1 plant was dwarf. And this gave us the phenotypic ratio 3:1. It's the way how Mendel conducted the experiments, right? He used pure plants. He ascertained as parents. That was known as parental generation. It took him 2 years to ascertain whether this plant, which he was going to select as parent, was pure plant. Then he studied inheritance of one character with its 2 alternatives at one time. He didn't use the complete inheritance of an organism. And when he performed other experiments with other 6 pairs of contrasting characters, 7 pairs of contrasting characters I have already told you, right? Out of which Mendel, in monohybrid cross, chose, height of the stem. And he got results in other 6 pairs of contrasting characters similar to this which he got with the character of height. In second F2 generation, tall and hybrid plants were obtained in 3:1 ratio. And from F2 generation, he raised F3 generation. And from F3 generation, he raised F4 generation. So on and so forth. But in every generation, he got a ratio 3:1. 3 plants were tall and 1 plant was dwarf. This is known as inheritance of one gene. It's the reason why Mendel, there were many reasons for Mendel, for his success. Because he did things in a systemic way. Other scientists took many characters into consideration at one time. They wanted to study the complete inheritance of an organism. Is the reason why they failed. But what Mendel did, he just did things in a very systematic way. He took one character with 2 alternatives, right? into consideration one time. Then he took 2 characters into consideration, dihybrid cross. Then he took 3 characters into consideration, trihybrid cross. Is the way how he succeeded, right? Let me tell you, tall plant and dwarf plant. These were used as parents. They were crossed. In F1 generation, all the plants were tall. Only difference between this tall and that tall was this tall and this tall was that this was pure tall and this was hybrid tall. Then these hybrid tall plants were self-pollinated in order to raise F2 generation. In F2 generation, the results vary. What? There were 3 plants tall, 1 plant was, dwarf. This is known as Mendelian phenotypic ratio. Monohybrid ratio. Sometimes it comes into exam. MCQ में आपको ये फायदामंद होगा 3:1. This is phenotypic ratio. What is the genotypic ratio of monohybrid cross? It's 1:2:1. 1 is the pure tall plant, 2 are the hybrid tall plants, 1 is the recessive plant. Okay? I hope that you might have enjoyed a lot. If you have enjoyed, like this video, share this video, and subscribe my channel, KD Sir's Botany Classes. Thank you. Till then, bye.